Good afternoon, everyone. So now we are going to have computer-aided drug design remedial class. In this particular class, how to attempt the questions in the sessional and what is the minimum answer which are expected to be present in the examination. If we are able to know it, we will not face any problem in the term end. First, what I would like to do is I will take each question from the sessional paper what we have got previously and what answers which are required to be present for each question. Briefly, I will discuss. Later onwards, I will share the documents to you and you can read, and we will have another remedial class where we can discuss the whatever the points which you are not able to understand further. Then we will go to the next topic and we will finish all our remedial classes. This is our strategy. And according to the first question, what he was given in the first session is identify the different stages of drug discovery and development. This is a 2 marks question. No need to write in the depth. So if you see the major 5 stages in the drug discovery and development, the first part is the discovery, where we are going to have target identification and validation, hit discovery and lead optimization. And then we are going to have a development of a molecule for selling in the market. That is going to be started with preclinical research. That is going to take safety and toxicity studies, ADMET also will be present. Then it is proceeding to the clinical research, phase 1, phase 2, phase 3, phase 4. So the phase 4 is actually the study where we are going to have for the post-marketing surveillance and where the safety monitoring will be done. And it is also as a part of pharmacovigilance we are going to do. In phase 3 is successful, we submit a new drug application or new biological license application, biologics license application for getting a approval to sell in the market. This is how our drug moiety is going to enter into the market. So the second question is what is define serendipitous drug discovery? It is the process of finding a new medicinal treatment by accident or a chance rather than through the pre-planned and systematic research programs. So if you see, the example is penicillin. You can give that is discovered by the Alexander Fleming. He noticed that a stray mold penicillin had accidentally contaminated a petri dish and killed the surrounding bacteria. That was the finding, which is resulted into a Sanjeevani-like drug for the most of the infections. The second thing is a Viagra, that is sildenafil, originally developed for the treatment of angina pectoris. During the trials, it was found that ineffective on the heart, but inducing the unexpected side effect like erectile dysfunction. So the erectile dysfunction can be treated by this moiety, which was observed as a side effect. Similarly, minoxidil, a oral treatment for the high blood pressure, has got a side effect where unexpectedly started growing excessive hair. So it is used for the repurposing for the baldness. Lithium, it is anti-maniac effect for the bipolar disorder discovered by the John CADD. While he was conducting the experiment on the guinea pigs based on the flaw theory about the uric acid. So this is repurposed later. Another question what we have got is the Lipinski rule of 5. In the Lipinski rule of 5, we have got one is molecular weight that is less than 500 Daltons. The second is log p value, which is less than 5. And hydrogen bond donors, not more than 5, that is less than 5. And hydrogen bond acceptor, not more than 10, that is 5 into 2, 10. So this rule of 5 make from the fact that all the numerical threshold for the multiples of the 5 is present most of the times in the drug-like moieties, and it is acting as a drug likeliness filter. The fourth question is application of Hammett constant and Taft constant. So these two constants are used. One is for the Hammett constant, which is measuring the electronic effect, both electron withdrawing or electron donating effect, and it is used in the QSCR. The Taft constant, it is used for the steric effect and electronic effect both. So this is the Taft constant can be determined by the hydrolysis of the ester predictions, where the size of the molecule is getting bigger. The ester rate, de-esterification rate will be decreased, and we are going to link this property to the steric effect, which is going to be present for the functional group. Similarly, we have got another question that is distinguished between SAR and QSAR. So SAR is qualitative approach and it is descriptive in nature. And QSAR is a mathematical approach. It is driven by the mathematical equation. We can predict the quantity. And this is the major difference. So where we are going to have in the SAR, replacing the OH by the Cl increases the activity or decreases what is going to happen. Whereas in the QSAR, we are going to find the activity in the form of equation, we can get the value. Similarly, we have got a bigger question where we have got comparative molecular field analysis. This will be taken separately in detailed manner, and comparative molecular similarity indices analysis. These two are the major one, which comparatively it has been given in the table. First, you go through them. We will discuss a little bit later in a complete manner. Another question which we have got is discuss in detail about the lead discovery and then analog based design. So in the lead discovery, we are going to search for a new chemical prototype. And while the analog is a systematic modification of that lead prototype to create a superior medicine. These are all the first is identification of the lead, second is improvement and modification of the lead. So this is present in the drug discovery as a one part. So in the lead discovery, we have got a random screening, non-random screening, and drug metabolism studies, and clinical observations. And later onwards, we are going to have an analog-based drug design, where we are going to have the analogs, which are having a resemblances to the lead, which can also lead into a drug-like moieties a little bit later, once we are successfully completed the study. So the another question what we were getting got in the first session is comparison of Hans analysis and free Wenzel analysis. So the Hans analysis is a physical-chemical properties where we are taking the log p value and sigma values and steric effect E constants, whereas free Wenzel analysis is the molecular structure, presence and absence of the groups. The Hans analysis is continuous and mathematical, whereas the free Wilson analysis is additive and it is also mathematically we can give as a equation. Needs the physical constant for the every substituent, needs the high number of analogs with varied substitution patterns. So this is the way it is having a difference. Whereas the Hans analysis can predict the activity of a new substituent not used in the study, but free Wilson analysis only can predict the combinations of the substances already tested in the set. What we have got in this particular case, both are predicting the why the drug works, whereas tells which group is the best for the specific position, it will work as a drug. Similarly, the formula is for the prediction of log 1/c, that is the minimum concentration which is required to produce the drug, which can be calculated by the hydrophobic constants and the electronic constant and steric effects. Whereas in the case of free Wilson analysis, we have got a mu and some of the contribution of the substituent for the biological activity where we are studying in the lab. There is a mixed approach which is also been suggested nowadays is Fujita band modification. It combines both the approaches. Using the Hans analysis, we can use it to map the pocket of the receptor. So by giving the equation, whereas the prevalence analysis, we identify the hotspots where we can have active sites to be predicted later onwards. So it will be resulting in the bonding with the drug receptor complex Now the another question what we have got in the exam is identify the factors applicable to the drug likeness screening, which is physicochemical properties, which includes molecular weight, log P value, hydrogen bond donor and hydrogen bond acceptor, polar surface area, and molar refractivity. These are all Lipinski rule of 5 and Weber's rule, which is being covered there. Structural and flexibility descriptors. There the rotatable bonds, that is Weber rule, and fraction of sp3 carbons which are present, and ring count and stereo centers. Admit and safety factors are also going to be working to be seen, which include solubility, metabolic stability, toxicophores, and permeability. Ligand efficiency matrix, which will be required to be considered is ligand efficiency, lipophilic ligand efficiency, and quantitative estimate of the drug likeliness. So that is having a single unitless score 0 to 1 that integrates the multiple physicochemical parameters into one index. So where the ligand efficiency potency related to the number of heavy atoms, and lipophilic ligand efficiency, that is potency related to the lipophilicity. So the log P value where we are going to have a linkage with the biological activity. The final question what we have got is explain the molecular docking procedures. So the procedure which includes the preparation of the molecule and target protein preparation. That is, we are going to pick up from the RCSB protein data bank, and then ligand preparation that we are going to selected from the databases like Zinc and PubChem, and active site identification and grid generation. So that is going to create a search space definition in the 3D grid box, and blind and site specific. If the binding site is unknown, the blind dotting approach, that is what the blind docking, which scans the entire protein surface through the site specific docking, and it will give the hotspots for the binding sites. Docking simulation, where we have got 3 types. One is rigid docking, and another one is a flexible docking, another is induced fit docking. Ultimately, at the end, we give the scores and evaluate their effectiveness. Ultimately, we validate the structure further. So the search algorithm, how the software move the molecule and score the function, is required to be seen in to find the steric effect and making the moiety fit in molecular dynamic situations. So where we are going to have a systematic method and stochastic methods. So here we are going to have such a finding a best pose that is going to be fitted, which is going to give the highest probability that will be working as a drug. The scoring function, it will be calculated by the energy also taken, most of the times Gibbs free energy. A more negative number means stronger the bond, which we need to take. Generally, we have got a force field analysis which calculate the physical forces like van der Waals forces and electrostatics. So constraints and challenges, we have to look into the water problem, where we have got a polar nature. Water contributes major in the drug receptor bonding. Protein flexibility, we need to consider a flexible only, not as a rigid protein. And the finalist problem, high docking score, does not always we can think that it is going to work as a drug. That is the reason we need to do the post-docking analysis. That is, look at the hydrogen bond networks, hydrophobic enclosures, and morpholodynamics to finalize the moiety. This is what we had got in the first sessional. I will open the floor for any questions.